Good afternoon. My name is Tim Gurman from Los Alamos National Laboratory. It's my pleasure to chair this afternoon's invited talk session. Our first talk will be given by Dr. Karissa Sanbonmatsu. Dr. Sanbonmatsu is a structural biologist at Los Alamos National Laboratory, where she leads the Sanbonmatsu Laboratory established in 2001. She received her BA in physics from Columbia University in 1992, her PhD in astrophysical, planetary, and atmospheric sciences from CU Boulder in 1997. She's been a leading figure in structural studies of long non-coding RNAs in epigenetics and is currently studying the mechanism of epigenetic effects involving chromatin architecture. She set the record for the largest biomolecular simulation at 2.64 million atoms in 2005, and again in 2019 at 1 billion atoms, publishing the first simulation of an entire gene. She's a fellow of the American Physical Society and Los Alamos National Laboratory, and an advocate for LGBT scientists. Her TED Talk, The Biology Agenda from DNA to the Brain, has over 2.5 million views on TED.com. The title of her talk is Supercomputing in the Biological Sciences Towards Zettascale and Yottascale Simulations. Please join me in welcoming Karissa.
Okay, I'm going to talk about supercomputing in the biological sciences and what we're doing out in Los Alamos National Labs to try to move toward the Zettascale and Yottascale levels of computing. Since the 1980s or so, there's been a movement in the biological sciences to see if we can simulate a biological system atom for atom. And if we have those simulations, can we learn something about mechanism and even develop new drugs? Well, if you think about it, this is a formidable task. Inside your body, there are trillions of cells, and in each one of those cells is an enormous amount of information. So in each one of your cells is your human genome, and that encodes billions of bits of information. This is encoded in DNA. DNA is a long string-like polymer, and that set of chromosomes in your cells contains about 3 billion repeating units, or nucleotides. So here we can look at just a tiny snippet of DNA, atom for atom. This happens to be just 8 nucleotides long. The sequence here is adenine, 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 guanine, guanine, guanine in the famous double helical form. And then if we look at an entire gene, and I'll explain how we developed this simulation later in the talk, we can see that it's quite complicated. So the DNA is organized very precisely in the nucleus of your cells. The double helical DNA is shown here in blue, and this is wrapped around about 2 times around an 8 protein bundle. Those units are called nucleosomes, and those are the kind of the cylindrical shapes you see here. And when the nucleosomes are in a more expansive conformation, they allow the transcriptional machinery to glom onto that and start expressing the gene. However, when it's in a more compact conformation, then it's less accessible, and you tend to get gene silencing. And so in terms of DNA, we want to understand this compaction and expansion and how that can upregulate or downregulate genes. And it turns out that these processes are critical for allowing you to do what you need to do to live each day and to have the thoughts you're having. They're also very important for taking a fertilized egg to an embryo to a newborn baby, and also are critical in many diseases such as cancer, controlling the gene switches that take a non-cancerous cell to a cancerous cell. Well, in all, to properly simulate, for example, the entire nucleus of a cell, you would need trillions of atoms to go for well over 10 to the 18 time steps. So that takes us way beyond even yottaflops to run. So the task ahead is formidable, but the positive side is this gives us a very clear path to well beyond yottaflops, very specific systems that we already have set up specific requirements in terms of memory, time scales, time steps, and performance, so we know what we need to do to get there in terms of the systems. Okay, so the simulations I'm talking about are molecular dynamics simulations, and there have been a lot of talks about molecular dynamics simulations at this meeting, so I think everyone knows what these are, but just to summarize, we solve Newton's equations of motion for a classical force field potential for every atom in our system, including surrounding ions and water, and then we advance atoms forward by 1 femtosecond, and then for each atom we have to include all the forces due to all the other atoms. And so, for example, if we just wanted to simulate only one gene, we need 1 billion atoms in that system. And then to get even 100 nanoseconds of physiological time, we have to repeat those calculations about 100 million times. And that's for just a few hundred nanoseconds. Now imagine going out to seconds or hours or days. Then the orders of magnitudes really pile up quickly. And then to go in a little bit more detail, this is what the typical pairwise interaction potential looks like between the atoms. We have harmonic terms representing bond vibrations, angles between 3 atoms, between 4 atoms, there's this dihedral angle, and then also van der Waals terms to prevent overlap. What's somewhat unique in biological systems is that these systems are highly charged, so you have to take into account long-range interactions, electrostatic effects, and these are done typically with a particle mesh Ewald algorithm that has an FFT in it. Okay, so what do we use molecular dynamics for in biosciences? Well, one of the main uses is to understand mechanism. We want to understand all the different molecular machines that are running around in your cells now that are reading the DNA, replicating DNA, doing chemical reactions, making the structures of the cell, building things, tearing things down. We want to understand how those work. Another more recent application is in integrative biology. So there you may have lots of different kinds of data. Some of the newest data is high resolution cryo-EM structural data. There's also single molecule FRET data, rapid kinetics data, mutational data, all kinds of different data. And the molecular dynamics simulations can sit in the middle of those pieces of data and kind of bring it together and present a coherent story and picture that summarizes all of the known experimental features of a system. There's also drug design, drug development, and so here we want to build or modify drugs and then try to predict binding to targets. There's vaccine design. We saw with COVID-19, there were several different mRNA vaccines, and you can use molecular dynamics simulations to examine and improve stability of the messenger RNA. And then finally, in biotech, there are many applications as well. So here we can see behind me is an example of integrative biology where we're using molecular simulations to look at the spike protein in the COVID-19 virus. And the gray envelope is a cryo-EM density, and the green structure is an atomistic model of the spike protein. And we can use the molecular simulations to get structures consistent with the cryo-EM density. And then in this purple, white, and green object is a molecular machine called the ribosome. And that's also important in vaccine design, in that the ribosome is what reads the messenger RNA that's shown in that green kind of squiggly polymer. Okay, so why should we use molecular dynamics instead of one of the other, or instead of sort of a more continuum approach? Well, often we can't really smooth out smaller length scales and time scales. The systems are often intrinsically discrete. For example, drugs bind to just one single nucleotide or one single residue of a system, so you can't smooth over the drug binding site, because that's often the key point on the complex that's used for drug design and drug studies. In addition, experimental data often consists of very small changes like point mutations. And there you're just changing one single unit of a system. And so these can't really be smoothed out. Chemical reactions, in terms of timescales, chemical reactions play heavily into processes, and these are at very fast timescales, such as femtoseconds. And then conformational changes happen rapidly also on the order of microseconds to milliseconds. So more reduced techniques, for example, coarse-grained or continuum models can give you a first look, but we would prefer, if we could, do full molecular dynamics simulations. And I just wanted to mention one caveat. In this talk, we're basically ignoring most of chemistry. There have been some amazing DFT and QMMM talks at this meeting, but here we're talking about mainly classical molecular dynamics, apart from a few rule-based chemistry processes we mimic. Now, some of the challenges with biomolecules are, as I mentioned, they can be inherently discrete, where phenomena at the atomistic level have important macroscopic implications. So for example, a single point mutation can cause sickle cell anemia. So that can have a huge effect, even though it's just one nucleotide, maybe 20 atoms in the system. There are many orders of magnitude you have to deal with. For length scales, in some cases as much as 10 orders of magnitude. So the chemical bonds are on the order of angstroms. The cells are on the order of microns, and you could even go out to meters for the sciatic nerve. And then for time scales, even more, 20 orders of magnitude. Chemical reactions on the order of femtoseconds, whereas chromosome rearrangements happen on the time scale of days. So there's a lot of orders of magnitude that we have to consider if we really want to do these detailed simulations. And in addition, as I mentioned, the systems are highly charged, so you need to cover long-range electrostatic interactions. And all in all, this requires many, many time steps, even to scratch the surface of being relevant. At minimum, 10 to the 9 time steps these days, but we'd prefer to go out way beyond 10 to the 18 or 10 to the 21 time steps if we had well beyond Yottascale platforms. But in the meantime, what we can do is we can leverage experimental data that exists and try to at least take a stab at these problems and see if we can learn something from simulations. So we could think of a holy grail for medicine of trying to do one human cell in atomistic detail with femtosecond time steps for 10 physiologically real days of time in your body. And this would require around 10 to the 18 exaflops or 10 to the 12 yottaflops. And so the goal of the talk I'm giving now is to try to get you to understand this slide in detail. So I'm going to show this at the very end, and then in the next several slides, I'm going to show a lot of different systems so you can more clearly understand this slide. But just briefly, in the lower left, we're showing a single protein. And this is kind of where we're at now. We can simulate a protein for around 1 millisecond to 10 milliseconds using the most compute power we can find and get, so this is maybe around 1 exaflop. And that's about what you need to simulate a protein, and that's about the relevant time scale of physiological time, maybe 10 milliseconds. Now for a nanomachine such as the ribosome or F1 ATPase, many different nanomachines, there we'd really want to go out to seconds, and so we're not there yet. There we would need maybe 1,000 exaflops to simulate the ribosome for 1 second. And then to go out to a virus or even a gene, we would have to go to much higher exaflops, 10 to the 9 exaflops. And the main reason is not only do these have a lot more atoms, but also it needs a lot more time steps, say 10 to the 16 steps or 10 seconds of physiological time. And then if we want to simulate a whole chromosome, there the time scales are much longer to be relevant. There we would need to go out to maybe 10 days. And so in that case, we would need many, many exaflops. And then finally, we can think about the nucleus and an entire cell. But the good thing is that we have very clear application systems getting us all the way out to the distant future in terms of HPC. We have a gene set up already, a chromosome set up already, a nucleus set up already. So we have these systems sitting there, but we just need a lot more cycles. Okay, so in the meantime, while we're waiting for these huge distant future platforms, what we can do is use experimental data to try to get around this time scale and time step problem and do more multi-scale integrated biology strategies. And so first I'll talk about some of the earlier work we did using experimental constraints to get around the timescale problem in targeted molecular dynamics on the molecular machine called the ribosome. So the ribosome is one of the key molecular machines, not just in your body, but in every organism that's ever been studied. In fact, in every organism that's been sequenced, the ribosome is almost identical from bacteria, plants, archaea, humans, all animals. The inner core is virtually identical in all those different systems. It's one of the most important molecular machines in the cell, and many think it was around at the times of the origin of life and very similar in those early days. And what it does is, what I'm trying to show here is a schematic cartoon picture of the ribosome. The ribosome consists of this yellow subunit and a blue subunit, and then it binds to this messenger RNA, the same kind of messenger RNA Moderna and Pfizer are working with for their vaccines, but these are your native messenger RNAs in your own cells. So it binds to that messenger RNA, and on that messenger RNA is the information that codes for a protein. So the information is residing in a gene in the DNA. It gets transcribed by RNA polymerase to the messenger RNA, and then the ribosome reads this messenger RNA, and then at each point in the process, there's a 3 nucleotide long codon, and then it looks for these transfer RNAs that have an anticodon, a 3 nucleotide anticodon, and it looks for something called Watson-Crick pair complementarity, where G's go with C's, A's go with U's for RNA. And these transfer RNAs are carrying these amino acids, and those are the building blocks of the protein. So it has to accept correct tRNAs, reject incorrect ones, and build piece by piece this protein. Okay, so this was almost 20 years ago now, where we were just doing what we could with, at that point, the Los Alamos Q machine back in those days. I believe that was 10 teraflops, I think. And so then we were looking at single barrier crossing events, but using a lot of experimental data to constrain the beginning and end points. We used a Lagrange multiplier constraint on the RMS to target at each step to look at a conformational change, and this required around 2.5 million atoms. And so here we can see the ribosome in atomistic detail, and I think I have to go back and then forward again. Okay, and in the bottom in purple is this small subunit, and in green is the messenger RNA. And then on the top is the large subunit in white, and we can see the simulation we did of the yellow tRNA moving into the ribosome. And this is not at physiological timescales, which would require a second. Here we were just doing around 10 nanoseconds, but at least we could see that kind of where the tRNA has to move and where it has to rub against the ribosome to get into its target state. From the cryo-EM experiments, we knew the beginning and end, and we essentially used molecular dynamics to interpolate between the two states. And then we could identify which specific nucleotides on the ribosome that the tRNA interacts with to get from point A to point B. And there we were able to make predictions about, if you mutated a certain nucleotide, would that have an effect? And we could also make predictions for which helices are important for developing new antibiotics. And so 2 groups validated this work. John Dinman's group in Maryland, and also the Ronina lab in Germany showed that the mutations of the nucleotides we predicted impair peptide release. Okay, so then next we can look at just free simulations, but here we're only going out to about a microsecond or so, so still it's well short of what you need to be really physiologically relevant, but it at least gives us a feeling of how much conformational space this molecular machine explores on the 1 microsecond time scale. And this also gives you a feeling of what the simulations look like. It's in a box of explicit waters, explicit ions, sodium, or potassium chloride, and magnesium. And then we can see how the ribosome moves when it's unrestrained. And it's mostly made up of RNA. The dark blue and the dark purple are RNA, a lot of different RNA helices folded onto each other. And then the curly Q things are proteins that also make up part of the ribosome complex. And this is one of the arms of the ribosome that does a lot of dynamics, as has been shown in single molecule experiments. And that helps recruit the tRNAs to the ribosome. And then in green is this piece of just a snippet of messenger RNA. And then if we look in more detail, we can see the transfer RNA in yellow and also the green messenger RNA. And this is this codon-anticodon interaction I was trying to describe. And then as we zoom out, we can see that the force fields in the MD simulations are very good. They give you rock-solid trajectories of the highly charged RNA molecules and the proteins, and the whole complex together just doesn't fly apart. And then the regions you expect to be dynamic are very dynamic. And this run we did in 2013, only out to 1 microsecond. So in this paper, we had a microsecond run and several hundred nanosecond runs. And we use this information to combine it with existing, the simulations were free, but afterwards, we combined it with existing measurements of kinetic rates. And from there, we could use the MD to estimate diffusion inside one basin to come up with a prefactor, and then from there get estimates of barrier heights. And then we put together these tables that if you go in the lab and you make a measurement of the rate, you could estimate a height from our MD diffusion estimates. Okay, so then the next thing we did is we looked at spontaneous transitions by the ribosome, again using experimental data as constraints, and also using a very simplified MD force field to get around the timescale problem. And this simplified force field is called a structure-based model, or an all-atom gold model. And so here, the native state potential uses this potential, the 0 points are defined by the native state, or in this case, the end state of this accommodation transition of the tRNA moving into the ribosome. And that enables us to basically have much faster times. These are not femtosecond timescales, but much bigger time steps. But we're able to use that because we have this native state force field potential. And so here we now can look at spontaneous transitions of the yellow tRNA that's trying to get into the middle of the ribosome. And so it's sampling around until it finds more favorable jumps that it can make, and then it moves to make these native state contacts. And then from this angle, we can also see the yellow tRNA moving toward its target. And we saw this in our targeted MD runs as well, that it interacts a lot with this helix 89. It moves toward the target and then back out, and then makes several attempts. And finally, when there's a favorable jump of this blue helix out of the way, the yellow tRNA can get into the middle. Here's from another angle. Here's this green snippet of messenger RNA, and we can see the tRNA sample all around and then finally make its way to the fully bound accommodated state. Okay. And then we would try to compare with single molecule FRET experiments from Scott Blanchard's group, who's at St. Jude's. And in single molecule FRET, just qualitatively we could see these recursive excursions of the tRNA trying to get to the bound state, and then it finally makes it there. And then one of the latest things we're trying to do, this is an even coarser level, even more smoothing, so it's mostly just rigid body motion of the entire ribosome. But we're just trying to get the feeling of what it would look like if we could simulate one of the tRNAs coming off the ribosome. We don't have enough computational power, and we won't have, this is something that would require 1 second, so we would need a zettaflop to do this run in atomistic detail. But this is an illustrative example where we have the system, we have an example trajectory already that we could, if we had a zettaflop of compute power, or 10 to the 3 exaflops, we could do this specific run in atomistic detail. Okay, so now I'm going to jump to a different kind of system. Those were molecular machines, which are a key class of things that are moving around in your cells to help your cells survive and do everything they need to do. Another class are genes and chromosomes, and that's another area we're pushing on at Los Alamos. And I'll talk about these simulations of a gene. This is related to the first movie I showed in the talk. And so to do this simulation of a gene, we started a collaboration with Tamar Schlick, who's kind of a powerhouse in chromosome modeling at NYU, and she's developed a lot of different coarse-grained methods to model genes and chromosomes, and chromatin, which is a region of a chromosome. And so she already had a nice, coarse, smooth model of a tiny region of a chromosome that's called a gene, or a gene locus, the region around the gene and the gene, called GATA4. And she made this by using experimental constraints. In those days, it was 3C capture, and did a quick Monte Carlo run to collapse a chain down using the experimental constraints. And she gave us a nice starting structure for this gene. And here she used her discrete surface charge optimization and rigid nucleosomes with some beads for the linker DNA and so forth, and the Monte Carlo. Then Cheng-Chung Tung at Los Alamos used his great algorithms to kind of make an all-atom rendering of that coarse, smooth model. So here he has nice algorithms to connect coarse-grained beads in all-atom form with the correct DNA helix geometries and twists and everything. And from that, we could thread that entire coarse-grained model with all the atoms of the DNA and the nucleosome proteins and so forth. And then we could come up with this first model of an entire gene. And then we collaborated with Yuji Sugita, and also all the folks who were putting the Trinity machine together at Los Alamos, and also people from Intel and Cray. And we made our all-atom explicit solvent, explicit water box. And then we used Yuji Sugita's code. He's at Riken on the Fugaku machine, but back then we were running on Trinity, and he and Jae-Woon Jung designed this Genesis code to scale extremely well. So we used their code and put this system on the machine and ran it a bit. Okay. And so this is, again, the GATA4 gene, and this is an important cardiac transcription factor. It's also important in embryo development for stomach and colon, and also for stomach and colon cancer. And it's an important target for epigenetic modification in heart development. And then for the MD, we used Yuji's Genesis code. It was explicit solvent, it was particle mesh Ewald. And in all, we had about 427 of those hockey puck shaped nucleosomes, and just over 1 billion atoms. And we got really nice scaling on Trinity with the Genesis code out to almost the full cluster that was called Trinity phase 2. And we ran for just a few nanoseconds. So even running this gene for just a few nanoseconds basically maxed out the Trinity platform. And we barely got to a few nanoseconds using everything we could at Los Alamos. So that's nanoseconds. Imagine now going to seconds, hours, or days. You know, there you can see how we can easily get to, you know, 10 to the 12 exaflops and so forth. Okay, and then again, I already showed this movie, but just now after explaining the simulations, we can see the blue double helical DNA wrapped around these 8 protein bundles, the nucleosomes, and we have over 400 nucleosomes. But this is a great intermediate system to go from a molecular machine out to a chromosome. The gene is something in between. It's about 1 billion atoms. And again, we're running just for 1 nanosecond, maxed out all of Trinity. Okay, so now I'm going to move on to another system. And in this case, we're trying to tackle looking at a full chromosome. So if we maxed out Trinity on just 1 gene, how could we even look at a chromosome? Well, what we do here is, again, we use our trick of leveraging experimental data. And in this case, we'll be doing quite a bit of smoothing. In fact, 1 bead in these simulations represents 1 billion atoms, so it's quite smooth. But we have tons of new experimental data that came out, so we can still learn something. And so what we're trying to do here is we're trying to look at how the X chromosome dramatically changes conformations, and also it creates RNA molecules that diffuse around the X chromosome and bind back to it. This is a process called X inactivation, and it turns out that you know, typical girls that are born have 2 X chromosomes, but 1 has to get fully inactivated for the baby to be viable. And that's the process we're studying here. Jeannie Lee is our main collaborator on this, and she's one of the leading experts on this system, and she's at MGH Harvard. So here, what we did is Anna LaPalla, who's now at Harvard also, developed a nice pipeline to generate 3D configurations of full chromosomes that are highly consistent with experimental data. And so in the last 5 years or so, a new technique called Hi-C came out with all the great high-throughput sequencing technology. And this has enabled people to create 3D reconstructions of chromosomes, the 3D geometry of the chromosome. And it essentially creates a contact map of the chromosome. You fix the cells with cross-linkers, and then you're able to get exactly the sequence locations of where the DNA crosses itself. So you might have seen on Google pictures of an X representing a chromosome. That's only at 1 very specific state during a cell cycle. In general, they're more globular. They look more like a ball of spaghetti. And they have many crossing points. So they cross each other in thousands and thousands of locations. With Hi-C, you can actually back out the exact sequence locations of where all those crossing points are. And then with Anna's pipeline, you can get 3D structures. Essentially, we run a polymer model, use virtual constraints corresponding to all the contact points, and get your structure. And then we can create a simulated contact map and compare experiment to simulation. And so here's 1 important state of X chromosome, the active state. And then here's comparing the simulation to the experiment. And we see we get really nice agreement. All the features are reproduced in the simulation that we see in the experiment. And then same for this other important state, which is the inactivated state of the X chromosome as well. And so then what we can do next is we can do our targeted MD trick and see what would a trajectory look like going from that active state to the inactive state, and try to look at the RNA diffusing around the chromosome at the same time. And so that's what we have here in this movie, where the gray is the X chromosome. The green are these RNA molecules that are diffusing around the chromosome, and then when they bind back to it, are the red. And so here we can study how the X chromosome moves from one state to the other, and how the RNA, one of the questions was, does the RNA penetrate through the middle of the chromosome, or does it go along the surface? We can see that the RNA can easily go into the middle and cover the full chromosome, which is what they see in the experiments. So that's one thing we learned from this. But really very important, and this is kind of the take-home message of this talk, is that this gives us a very clear pathway out to well past yottaflops, to very futuristic platforms, because we already have a simulation done at a very coarse level, but of a system that would require that much compute power. If we could just render this in all atom form and go for 10 to the 21 time steps, then we'd have it in atomistic detail. So it gives a very clear path forward to go to maybe 20, 30, 40, 50 years now. What kind of platforms, what problems would we run on those platforms? And so I'm just going to start to wrap up here, where we can now think about the chart I showed in the beginning of the talk, but now plotting some numbers of where we are now. So for this small protein system, we pretty much have this. D.E. Shaw's group, they have their MD specialized computer called Anton, and they can run 1 or nowadays I think even 10 milliseconds on a protein, which is pretty much all we need. So we're there just about for a single protein. As an aside, Google AlphaFold also just does 1 protein but with the 0 time steps. So we're there in terms of what we need for a protein. For a nanomachine, for example, like the ribosome, we're at around microseconds now. So we still have to go, we still need a lot more to get to a 1-second simulation of the ribosome. A gene, we're even more far off, where we're only at a few nanoseconds, maxing out something like a Trinity at Los Alamos, where we'd like to be way up here. And then we can't even really have 1 step of a chromosome yet, because we don't have memory to fit, say, 100 trillion atoms there and do electrostatic calculations. So now I'll show the same chart I showed in the beginning, where we're there at an exaflop for 1 protein, but to do a gene, we would need more like 10 to the 9 exaflops to do all those time steps for 1 billion atoms. And then for a chromosome, remember in this chromosome simulation, 1 bead was 1 billion atoms. So there, to get all the time steps, we would need more like 10 to the 15 exaflops, and then for a whole cell, more like 10 to the 18 exaflops. But again, this gives us a very clear pathway where we have the systems just sitting there, and if we had some futuristic platforms, we'd be almost ready to go. So to conclude, I want to emphasize that molecular dynamics, MD simulation, is one of the main techniques used in the biosciences for drug development, biotech, and mechanistic understanding. Length scales, electrostatics are important, and there's a large range of length scales you need to cover, like the point mutations. Time scales are even more so. We need femtosecond time steps, but we'd like to go out to day-long simulations. And so the upshot is that simulating a whole cell for 10 days would require 10 to the 12th yottaflops. Okay, so I'd like to thank especially the agencies who've supported us over many years, NIH, NSF, DOE, especially Los Alamos Institutional Computing, who's been so generous to us over all the years, all of my great collaborators, and all of my great postdocs, and you for your attention.
Okay, thank you, Karissa. We have time for a couple of questions. I believe we have one in the back.
Yeah, good afternoon. Thank you for this very nice and impressive talk. I'm Claude Tadonki from Mines Paris Tech Institute. The flops that you mentioned, I guess, was based on the theoretical flops of the machine, the potential. But what is the sustained flops that you get in your simulation?
The sustained versus the theoretical, I think, is the question. Yeah, these estimates I was using were based on actual sustained performance, but there were only a few points, like basically the Trinity run, a Q machine run, a few, and then I did a lot of extrapolation, I'd say. So I'd say there's room for a few orders of magnitude different, but it would be on the level of, say, 10 to the 7 or 10 to the 17 or 10 to the 18 exabytes. It might be a lot more because we ignored chemistry here, and also I was assuming a linear order n scaling when it should be more like order n log n for the number of atoms, so it could be even more. But, okay.
Razvan Marinescu from UC Santa Cruz.
Yeah.
What advances in hardware and software do you think we need to get there?
Oh, that's a great.
Make some guesses.
Yeah, I don't know. I mean, if you could get quantum computers so that you could use them for a problem like this, that might help. But the more immediate problem right now is on GPU scaling versus nodes, because right now, at least in biomolecular simulation, we're at this point where you don't know if it's better to run on a few GPUs or on a large cluster. It's almost the same. If we could somehow leverage the GPU power and have it scale, that would get us, that would be a great jump forward, I think.
And do you think machine learning could play a role here?
Machine learning is being used a lot in the quantum mechanical side of things. For QMMM, they call it MLMM, and it's been successful there, but to go to these large systems, there it's a lot more challenging to do the more classical style MD, yeah.
Thank you, thank you.
Good, let's go over there.
So in your gene simulation, could you modify the acetylation state of the histones, or was that just to let it ride?
So I didn't quite get
in the chromatin simulation?
Yeah.
Were the histones acetylated and methylated?
Oh, I see, yeah. No, this is kind of just the 0th order simplest system that we could set up, but that's where we're going next. And actually in the chromosome simulation, which is very coarse-grained, there we're already doing a little bit with having kind of modified beads and so forth. But yeah, that's one of our next steps for sure.
And what force field did you use for the ribosome? Because most of them are terrible, especially.
Yeah, we use Amber, and there are a lot of new RNA Amber modifications out that work quite well, but we think that the very careful equilibration of the ionic environment we find is so important to get nice stable trajectories for RNA. So we do a lot on that.
Let me step in a minute. We have a question online.
Okay.
So thank you for the nice talk. You mentioned AlphaFold. In your opinion, what role will such AI-powered systems play in achieving the goals you describe?
Sorry, I didn't catch the end of that.
What role will AI-powered systems play related to the previous?
I see. So for AI, yeah, I think AI is making a lot of advances for the quantum calculations, and for classical, there's still a long ways to go, but it means there's a lot of opportunity there, I guess.
Okay.
First of all, thank you for your beautiful presentation. In a previous life, I also was in molecular dynamics, and I remember this feeling of feeling constantly under threat from a variety of fronts, right? One front is force fields, tiered MD, target NMD have a lot of detractors. They say it's not real science, it's an approximation. The ground truth is only the quantum truth or the wet lab truth, right? Reconciling with wet lab truth is expensive, it's problematic, it's difficult to do. Now machine learning and AlphaFold are another threat. And then finally, as you displayed so well, fully atomistic MD at interesting timescales has a hunger for compute that is basically beyond planetary scale. Like if we used every atom of silicon to make computers on Earth, probably we wouldn't still get to the threshold. So do you also feel that the field is under so many threats? And how do you see things being in 5 years? Like at Supercomputing 2027, if you show up again, will you tell us fully atomistic AMD is dead and it's being killed by ABC, whatever technology it is, or it still survives, or it has done enormous breakthroughs that we didn't expect? How do you see it?
I'm very optimistic. I used to be very pessimistic, but I think things are really shifting. Number one, with the experimentalists, there's been a sea change in the structural biology where cryo-EM has really displaced X-ray crystallography. So now it's all about cryo-EM, and they're much friendlier to MD folks. They were often working in the same lab together. We develop algorithms together. And also the cryo-EM data has dynamics in it, so they're interested in using the MD to see the dynamics. So that's helping quite a bit. And also I don't see AlphaFold as pressure. I think it's building more awareness of structural biology, and I think in atomistic modeling, I think that helps as well. But in terms of molecular dynamic simulations in general, I see it in the 5 years as maybe being an industry driver where we need faster computers to do these calculations. We can only do one protein now, and so that doesn't get you very far in terms of making a blockbuster drug. You know, we'd like to do a whole complex or a membrane protein or something like that. So we need much bigger platforms to go deeper into these problems, I think. Yeah.
Okay, we're out of time. Please join me in thanking Dr. Sanbonmatsu.